Welcome to GNANA, a concept-based digital learning. I'm Dr. Swarupa, lecturer in microbiology from Telangana Social Welfare Residential Degree College for Women, Mahendra Hills. Water is a basic human need. Indeed, it is essential to all forms of life. If there was no water, there would not be any life on earth. Everyone has the right to sufficient, continuous, safe and acceptable water for personal and domestic use. However, millions of people in India and other countries are suffering from water scarcity. Safe water, sanitation and hygiene, collectively known as wash services, are crucial for human health and well-being. When you look at the statistics of progress on wash services, according to WHO UNICEF 2017, it was reported that 785 million people lack even a basic drinking water service and 144 million people still dependent on surface water, though there was a slight improvement from 2015. Globally, at least 2 billion people use a drinking water source contaminated with feces. And 10% of global population spend more than 30 minutes per round trip to collect water. Further, 673 million people still practices open defecation and 2 out of 5 people still lack basic hand washing facilities with soap and water at home. Moreover, it is estimated that by 2025, half of the world's population will be living in water-stressed areas. Despite several efforts by government at various levels, still there are many people living in water-scarce regions and dependent on polluted water, mainly in developing countries. Polluted or contaminated water and poor sanitation is the predominant source of waterborne diseases that are a major cause of morbidity and mortality. Today, waterborne diseases are a common cause of illness in India as millions of people depend on river even if it is carrying a polluted water. Climate changes like severe rainfall, floods, rise in sea level, high temperatures and drought conditions promote the outbreak of waterborne diseases. Though the waterborne diseases occur throughout the year, we can notice the peak during seasonal changes in summer, monsoon and post-monsoon period. It is important to understand what are the causative agents responsible for the incidence of waterborne diseases. So, our topic for today is waterborne pathogens and their transmission. In this session, we are going to learn about what are waterborne diseases and their contribution to the total disease burden and some of the potential waterborne pathogens of public health importance, their disease transmission and preventive measures. Let's have a look at what are waterborne diseases. Waterborne diseases are those that are transmitted through ingestion of water contaminated by pathogenic microorganisms such as bacteria, viruses, protozoa and helminthic parasites. World Health Organization estimated the contribution of water, sanitation and hygiene related diseases on the total disease burden. Overall, 1.9 million deaths and 123 million disability adjusted life years has been reported from inadequate wash services, which accounts for 3.3% of global death, 4.6% of dailies and 13% of death among children under 5 years. Here, dailies means disability adjusted life years, which measures the years of life lost to premature mortality and to disability. It is clearly evident from this image that diarrheal diseases account for 41% of the total disease burden and it is the second leading cause of death among children aged under 5 years. Also, the largest contributor to waterborne disease burden. It has been estimated that around 8,29,000 people die each year globally from diarrheal disease due to unsafe drinking water, sanitation, and hygiene. Let's quickly move on to the waterborne pathogens. There are many bacterial, viral, protozoal, and helminthic pathogens responsible for waterborne diseases. Among these, important bacterial pathogens include Vibrio cholera, Salmonella, and Shigella species, E. coli species, Burkholderia, Campylobacter, Francisella, Legionella species, and Mycobacterium avium complex. While viral pathogens include hepatitis A, hepatitis E, rotavirus, adenovirus, astrovirus, norovirus, and sapovirus. Other waterborne protozoan and helminthic parasites comprises Giardia intestinalis, Entamoeba histolytica, Cryptosporidium species, Toxoplasma gondii, Dracunculus medinensis, and Fasciola species. Majority of waterborne diseases spread through fecal-oral route of transmission, wherein the pathogens present in the human excreta transmit from one host to the other through a variety of means. This F diagram shows that the pathogens present in the feces of one person travels through the fingers, flies, fields, fluids, and food to the mouth of the other person. Most of the fecal-oral route of diseases occur through the ingestion of contaminated water or food and less often through inhalation. Main causes for fecal-oral route of disease transmission is due to open defecation, poor sanitation, and hygiene practices. Most of the waterborne pathogens infect gastrointestinal tract and results in diarrheal disease along with other common symptoms such as fever, nausea, vomiting, and abdominal cramp. Among these, diarrhea is the principal cause of many child deaths. Majority of diarrhea cases result from two important pathogens such as rotavirus and E. coli, and others include Cryptosporidium and Shigella. Diarrhea is passing of loose watery stools 3 or more times a day, which results in dehydration and if untreated even leads to death. Let us now look at some of the bacterial waterborne pathogens. Vibrio cholerae is a curved, rod-shaped bacteria that causes acute diarrheal disease. It is a gram-negative and high motile bacteria with a single polar flagellum. The motility of Vibrio cholera in stool samples from infected patients resembles fish in stream appearance. And there are two antigens present, one in flagella called H antigen and other in the cell wall lipopolysaccharide layer called O antigen. Based on the somatic antigen, Vibrios are classified into subgroups called TSWREIS from O1 to O139. O1 and O139 are responsible for many epidemics of cholera. A person can acquire the infection by ingestion of water or food contaminated by fecal matter. It has been shown that 10 to the power of 3 vibrios are enough to cause a successful infection. Upon ingestion, the vibrios adhere and multiply on intestinal epithelial cells, secreting an enterotoxin called choleragen or cholera toxin. This toxin production is largely limited to vibrios, which are lysogens harboring the DNA of the filamentous bacteriophage called CTX5. The severity of the disease is due to the production of cholera toxin. The toxin consists of 5 binding subunits and 1 active subunit A. The B subunits attach to the GM1 ganglioside receptors on the surface of jejunal epithelial cell, releasing the A subunit inside the enterocytes. The A subunit splits into A1 and A. The A1 fragment is active toxin that causes prolonged activation of cellular adenylate cyclase and accumulation of cyclic AMP. This results in outpouring of large quantities of water and electrolyte into the intestinal lumen, leading to diarrhea. It takes between 12 hours and 5 days for a person to show symptoms after ingesting contaminated food or water. Clinical features of cholera are mainly due to massive loss of fluids and electrolytes. The common symptoms usually include diarrhea, nausea, dehydration, and muscle cramps. In 10% of cases, profuse watery diarrhea and vomiting may lead to hypovolemic shock and death. A typical symptom of cholera is colorless, watery stool with flecks of mucus and a characteristic sweetish odor. The stool resembles water in which rice has been washed, hence called rice watery stools. Cholera is an endemic disease in Southeast Asia and especially India. Cholera outbreaks have occurred on a frequent basis in the past. Cholera outbreaks are common in our country during floods or pilgrimage where large number of people assemble. During 2019, around 0.9 million cases, 1900 deaths were notified from 31 countries globally. Diagnosis of cholera involve testing the stool sample or rectal swabs for the presence of Vibrio cholerae through microscopy, culture, biochemical and serological analysis. Prophylactic oral vaccines for cholera have been tried without any promising results. Hence, no ideal vaccine is available as yet. Improvements in water and sanitation are critical in preventing cholera outbreaks. Treatment of cholera involves prompt administration of oral rehydration solution to replace the lost fluid and electrolytes. Antibiotic therapy is of secondary importance. If urgency arises, tetracycline is an effective drug. In October 2017, Global Task Force on Cholera Control launched a strategy that aims to reduce cholera death by 90% and to eliminate cholera in as many as 20 countries by 2030. Let's look at another bacterial waterborne pathogen, typhoid-causing bacteria, Salmonella enterica typhi. Salmonella typhi is a rod-shaped gram-negative bacteria with peritrichous flagella. The infection is acquired through consumption of water or food contaminated with feces of an infected person. The Salmonella possess flagellar H antigen, the cell wall O antigen, and a surface polysaccharide Vi antigen. Once ingested, the bacilli attach to microvilli of ileal mucosa and penetrate into the submucosa where they are phagocytosed by polymorpholeucocyte and transported to the mesenteric lymph nodes. The bacilli multiply and invade the bloodstream via the thoracic duct. In 7 to 10 days, transient bacteremia ensures seeding the bacilli in liver, gallbladder, spleen, bone marrow, and kidneys. The bacilli multiply further in these organs and again pass into blood, causing secondary bacteremia. The bacilli are discharged from gallbladder continuously into the ileum, resulting in characteristic typhoid ulcers. Symptoms include high fever, also called step-ladder pyrexia, headache, stomach pain, weakness, vomiting, and loose stools. Raw spots appear on skin in second week of infection. Severe complications can include intestinal perforation, hemorrhage, and circulatory collapse. Typhoid fever occurs most often in children and young adults between 5 and 19 years of old. Outbreaks of typhoid fever are frequently reported from sub-Saharan Africa and countries in Southeast Asia. In the United States, about 400 cases occur each year, and 75% of these are acquired while traveling internationally. In typhoid, convalescent carriers are more common, about 10%, and chronic carrier status exists in about 2 to 4%. The most notorious carrier of typhoid fever in history has been Mary Mallon, known as Typhoid Mary. In 1907, she became the first carrier in the United States to be identified and traced. She was cook in New York who was associated with 7 outbreaks and over 200 cases. Diagnosis is made by testing blood, stool, bone marrow, or urine samples by culturing. Major diagnostic test is Vidal test that detects antibodies against Salmonella somatic O and Flagella H antigens. The test also is used to estimate the antibody titer values in typhoid fever. Specific prophylaxis is by vaccination. There are 3 types of vaccines available. Oldest is the heat-killed tab vaccine, an injectable vaccine, Typhoral, based on the purified VI antigen for people aged over 2 years, a live attenuated oral vaccine for people aged over 5 years. Effective antibacterial therapy include administration of ampicillin, ciprofloxacin, azithromycin, and cephalosporins. Now, let's have a look at the few important viral pathogens responsible for waterborne diseases. Hepatitis A virus is one among the heterogeneous group of hepatitis viruses which spread through fecal-oral route or sometimes direct contact through an infected person. It was initially designated as enterovirus 72, which measures about 27 nanometers, and it is non-enveloped or naked positive single-stranded RNA virus with icosahedral symmetry belonging to the picornavirus family. Infectious hepatitis or type A hepatitis is a mild to severe liver disease caused by the ingestion of hepatitis A virus. After ingestion, virus multiplies in the intestinal epithelium and reaches to the liver for further multiplication by hematogenous spread. Virus sheds in the feces and followed by development of viremia. And incubation period for this is the 2 to 6 weeks. Type A hepatitis affects mainly children and young adults, and majority of them are asymptomatic. If a person develops symptoms, disease occurs in 2 stages. In the prodromal stage, symptoms begin with fever, malaise, anorexia, nausea, vomiting, and liver tenderness. These symptoms usually subside once the jaundice develops in the icteric stage. Type A hepatitis occurs worldwide sporadically or as epidemics. According to WHO, hepatitis A responsible for 7,134 deaths, which accounts for 0.5% mortality. In India, it is the most common cause of acute hepatitis in children, and by the age of 10, 90% of population will acquire infection. Diagnosis of hepatitis can be done by either direct visualization of the fecal sample by immunoelectron microscopy or by detecting antibodies by enzyme-linked immunosorbent assay. In addition to the general preventive measures, a 2-dose inactivated vaccine is used in many countries, and it is recommended for the people who travel to the countries where the virus is endemic and other high-risk group people. Natural infection with hepatitis virus provides lifelong immunity, and treatment is symptomatic and no specific drug is available. Moving on to the next most important waterborne pathogen, rotavirus. It is a double-walled virus with a characteristic wheel-like appearance. The genome of rotavirus is double-stranded RNA with 11 segments. When you look at the pathogenesis of rotavirus, it enters into the body through the ingestion and infect the cells in the villi of the small intestine. Then they multiply within the enterocytes and alter the transport mechanisms, which in turn prevent the absorption of water, causing excessive secretion of water and loss of ions, result in watery diarrhea. The typical, as seen in the epidemiology, rotavirus occurs throughout the world, but India is the most affected country. Rotaviruses are one of the most common causes of severe diarrhea or gastroenteritis in infants and young children. By the age of 5, nearly every child will get infected. According to the Centers for Disease Control and Prevention, it is responsible for up to 5 lakh deaths each year worldwide. The typical symptoms of rotavirus gastroenteritis include watery diarrhea, fever, abdominal pain, and vomiting, leading to dehydration. Vomiting is usually of short duration and may occur before or after the onset of diarrhea. Stools are usually in greenish yellow or pale in appearance. Rotavirus diarrhea is self-limiting and patients recover completely within 5 to 10 days. The gastrointestinal symptoms generally resolve in 3 to 7 days. Incubation period for this is 2 to 3 days. Laboratory diagnosis of rotavirus infection can be done by microscopy, serological, and molecular tests. There are 3 World Health Organization approved live attenuated oral vaccines available for rotavirus prophylaxis: Rotatec, Rotarix, and Rotavac. The usual treatment of choice is oral rehydration with fluids and electrolytes, and if required, intravenous rehydration therapy. In selected clinical cases, antirotaviral immunoglobulin therapy is followed. Let's now discuss briefly on some of the waterborne protozoal parasites. Giardiasis is a diarrheal disease caused by the microscopic parasite Giardia lamblia. Once a person has been infected with Giardia, the parasite lives in the intestines and is passed in stool. Once outside the body, Giardia can survive for weeks or even months. The most common symptoms of giardiasis include diarrhea, foul-smelling stools, and abdominal cramp. Amoebiasis is caused by the parasite Entamoeba histolytica. Amoebiasis or amoebic dysentery is more closely related to poor sanitation and socio-economic status than to climate. The parasite exists in 2 forms: vegetative form called trophozoite and cystic form. Trophozoites multiply and encyst in the colon. The infective cysts are excreted in stool and remain viable for several days in feces, water, sewage, and soil. The clinical symptoms of amoebiasis range from diarrhea and dysentery to fulminant colitis and peritonitis. Acute amoebiasis can present as diarrhea or dysentery with frequent, small, and often bloody stools. The other protozoan parasite, Cryptosporidium parvum, causes a diarrheal disease called cryptosporidiasis. The small intestine is the most commonly affected site by Cryptosporidium, along with the digestive tract or the respiratory tract. Cryptosporiasis is an intestinal illness caused by the microscopic parasite called Cyclospora. People become infected with Cyclospora by ingesting water or food contaminated with spore-related oozes, which are the infective form of the parasite. Finally, Balantidium coli is the only protozoan ciliate infecting humans, causing the disease Balantidiasis. Swine is the primary reservoir host and cyst is the infective stage. For the treatment of protozoal diseases, apart from oral dehydration, the usual drug of choice is metronidazole, timidazole, albendazole, and emetic, etc. The important waterborne pathogens, the disease caused, and their mode of transmission is listed here. Bacterial pathogens and their diseases, viral, protozoal, and helminthic pathogens and their diseases. Most of them are transmitted by fecal-oral route. Let's have a quick glance at general preventive measures of waterborne diseases. We can prevent the spread of waterborne diseases by ensuring the access to safe water, sanitation, and hygiene services, by improving the quality of drinking water, by following the good food hygiene, avoiding open defecation, and by practicing safe disposal of waste, human and animal excreta, and washing hands before eating. The current COVID-19 pandemic taught us the importance of washing and sanitizing hands in preventing the spread of not only coronavirus but many other infections. In addition to the individual practices, United Nations has come up with the Global Action Plan to ensure universal access to safe and affordable drinking water and adequate sanitation for all by 2030 as one of the Sustainable Development Goals. Let's recap what we have learned today. Waterborne diseases are infections that are transmitted through the drinking of contaminated water with microbes and contributes major portion to the total disease burden. Most of the waterborne diseases spread through fecal-oral route, and we have seen the potential waterborne bacterial and viral pathogens such as Vibrio cholera causing cholera, Salmonella typhi causing typhoid, hepatitis A causing viral type A hepatitis or infectious hepatitis, and rotavirus causing diarrhea, and even the protozoal pathogens and their diagnosis, treatment, and preventive measures. Hope we all save water for future use and follow good hygiene practices to prevent deadly diseases. Safer water, better health. Thank you.